So I'm Dejan. I'm co-founder and CEO of denovoMATRIX, and today's talk would be different from all the talks that you've heard so far, in which most of it has been about understanding the extracellular matrix of what we have in nature, right? With denovoMATRIX, what we're trying to do is a little bit of the opposite. We're trying to take the lessons learned from nature and building them for our purposes. And specifically, the purpose which we have is building and using ECM for culturing, for cell manufacturing, so culturing cells, but specifically stem cells, for uses in things like cell therapies or in very futuristic applications like cultivated meat. So today I'll be talking about process and cell-specific biomimetic matrices and how they enable the culture of functional stem cells. So the goal that denovoMATRIX has specifically is to enable cell manufacturing. For us, it's specific to stem cells, and particularly to enable it across different scales. And I'll talk more about that because you have scales that run from sort of laboratory all the way through industrial. Shortening development times. This is a big issue in the industry, is you have always development for various projects, various processes, various cell types, various disease indications. And finally, reducing costs. So cost of goods, the manufacturing behind cells is the biggest hindering factor behind having more applicable and more readily available therapies for patients. So coming to the first part, cell manufacturing across scales, what does that word actually mean, and what does it look like? So typically what you have in a cell manufacturing process is you start off with an isolation. So it means you either start off with patient material, animal material, and you isolate out the cells that you're looking to expand. So you start off with a heterogeneous mixture. So for example, if you're talking about a patient, you would start off with a bone marrow puncture, which then gives you heterogeneous mixture of different cells. You have blood cells in there, you have muscle, you probably have some fibroblasts, and you also have stem cells, which is actually what you're looking for. So your first step is typically isolation. So that's where you're trying to pull out from this mixture a single cell type of interest that you're looking to expand. Moving beyond that, you have an expansion phase where you're looking to grow more of this particular cell type, but also to characterize it. Do I really have what I'm looking for? Is it really what I want? Are these cells still functional for my end purpose? And then the last stage, which is then upscale and the actual therapeutic application. So that means, am I growing it to the quantities that I need to actually treat a patient for the number of patients which I'm trying to treat in my particular study? Now, you need biomatrices for a lot of these different aspects, and they are important for different purposes. For example, for enhancing the proliferation of these cells. Sometimes cells are not very happy to grow outside of the body. They are growing at very slow rates or not at all. You want to increase the total yield at the end of your process, but you also want to increase the stability of these cells, so they inevitably change over time as you grow them. And finally, you want to have a certain functionality of these cells. So it doesn't help if you grow a huge amount of cells to use them in a therapy if they don't have that functional output, if they can't perform the end job that they need to perform. So today I'm going to dive deeper into a particular use case, which is for mesenchymal stromal cells. This is a very well-known cell type for one of the first cell therapies out there, where they would use mesenchymal stromal cells to treat particularly immune diseases, and how we have developed biomatrices specifically for different scales of growing mesenchymal stromal cells and applying them finally for patient use. So what I think is an interesting factor here is that you face a big problem both in the isolation and in the expansion of these particular cells. So the traditional way is, as we said, we start off with patient who typically undergoes a bone marrow puncture procedure, and then you have to isolate out MSCs from this mixture of cells of bone marrow. And this is a procedure which is not always a given, so sometimes the patient will have to undergo multiple punctures to get sufficient stem cells isolated at the very beginning of this process. At a later stage, you would then take these MSCs that you've isolated and you would expand them in a laboratory scale, so in small 6-well plates, tissue culture flasks, and then eventually, if you're looking to treat not that one patient, but you're looking to treat a multitude of patients, let's say 10, 20, 50, 100, from the same source, you would then move to something like a bioreactor setup, where you have this impeller, you have multiple MSCs growing in a big solution. So how do we address these problems, and how do we use extracellular matrix in order to solve these big issues? So denovoMATRIX came from originally from the B-Cube, so we are a spin-out from this institute, and we owe a lot of our knowledge about extracellular matrix based on what we've taken from our group, from the group of Professor Zhang. And what we've created is what you see here, the screen matrix. So the technology behind denovoMATRIX is essentially a biomaterial that combines two of the key factors of the extracellular matrix found in mammalian systems. Specifically, these are the proteinaceous aspect and the sugar-based aspect, so glycosaminoglycans specifically. And what we've created is something we call the screen matrix. This is one of our first products and is now a screening service which our company offers. And what it does is essentially combines various glycosaminoglycans found in the body with various peptides which mimic the proteinaceous aspect of the extracellular matrix and create such a screen, right? So what you're looking at is a 96 well plate with 96 different combinations of glycosaminoglycans and peptides which mimic the various extracellular matrix proteins found in the body. So if we look at some of these examples here, you see typical adhesion proteins found in ECM like fibronectin, laminin, vitronectin, but you also find somehow more structural proteins like collagen, which is a big part of your bones, hair, nails, as well as other proteins which are not necessarily responsible for adhesion, but for some kind of actuation of cells. So we're talking about cytokines, growth factors. So these are all, this is what we just termed under diverse, because it can really be a lot of different things. So the idea here being that we can have a chemically defined system which mimics the natural ECM and allows us to pick out and find an optimized extracellular matrix for the particular job which we're looking for. So using what we started with, mesenchymal stromal cells, we want to find a ECM which is optimized for isolating these cells in the first step, and in a second step, finding an ECM which is optimized for expanding these cells in a functional way. So what you're looking at here is the first screen, how typically we start off with all projects involving the screen matrix. You would seed your cells or your mixture of cells into the screen matrix with all of its different types of coatings, and then you would start to see a sort of pattern or fingerprint start to appear. So we have some ECMs which are totally blank. So this is a microscopy image. What you're seeing stained in green are the cells. So some of them are completely blank, so cells don't survive and end up dying and lifting off. And in other examples, you have a very dense carpet of cells starting to grow and proliferate, indicating that these are the ECMs which we want, and the ones here are not the ones which we want. All right, so we start off with this screen of different ECMs, and then we start to narrow them down by eliminating as many as we can until we can find the optimized one, which is good for the particular purpose we're looking for. So we published a paper last year in Cytotherapy where we set ourselves this exact goal of isolating MSCs from patients using a chemically defined or serum-free method. And what you see here are some of the data which we've managed to collect from that and comparing it to a traditional method of isolating cells. So I mentioned to you at the beginning, some patients have to go through multiple rounds of bone marrow puncture in order to get something because you don't have enough cells. So this is what you see here is some comparisons to the sort of standard. Here we call the standard is tissue culture plastic. And you can see that the ECM which we have developed using the screen matrix method, we call isomatrix, and it enables us to get up to 50% more cells by day 4, and then the comparison starts to grow much, much larger by day 11 to 14, and then in the final end step, it grows even larger than that, right? So as your cells proliferate, then differences also tend to grow more quickly. So just as an example what the cells look like, this is a typical image, phase contrast image of MSCs on a surface quite dense. And this is on isomatrix. You have this dense carpet of cells that are isolated and are growing well versus tissue culture plastic, which typically have lower amounts of cells isolated. Another interesting aspect of the matrix we've identified is that when you look at the percentage of viable cells and their cell size, you start to see differences and you start to see these peaks in generally populations of smaller cells. And in previous literature, this also means that these cells tend to be have a higher stemness, higher proliferative capacity, than cells which tend to be larger or have a more amorphous mixture of sizes. So we have some interesting evidence already at the beginning showing us that isomatrix is helping with the isolation of these MSCs. So more information which we collected, so this is some more data from the paper, is looking at functionality. I've told you it's important to understand the cells which are being expanded, are they functional at the end? Can they really treat a patient? And a way that we can test that is we can look at various functional tests for these MSCs. Are they able to have the capacity to differentiate? That's why we run this so-called trilineage differentiation, where they're differentiated into bone, fat, and cartilage tissue. And we see basically positive signals for those three. You have so-called colony-forming unit assay, where you seed these cells at extremely low concentrations, and you see whether they are able to survive sort of adversity and form so-called colonies. You see also drastic differences between the sort of classical method of isolating and the isomatrix isolated cells. So they seem to be able to pull out more stem cells, true stem cells, than just proliferators using this method. And in the end here is a true test of functionality for one of the most common uses of MSCs. So MSCs are used to treat something called graft versus host disease. So this is a disease where typically a patient will have a very, very high immune response to graft tissue that they've received. And this means that they have to be on immunomodulatory drugs for the rest of their lives. And MSCs can be used from the same patient to basically calm down this immune response and bring them back to a sort of stasis without having to use immune modifying drugs. And this test here that you see on the far right is exactly showing that. So you would typically take it's a test involving basically immune cells and MSCs. And what you see here with these four donors is the comparison to a no MSC, which basically produces your highest proliferation response. And with MSCs grown on our isomatrix, you can see this reduced response, this capability to modulate the immune system. So I've talked about isolation, that was the topic of our paper, but then what happens after you've isolated the cells? So that's the first step. The next step is how do you expand them, and what are the conditions in which you expand your cells? So MSCs can be expanded in a variety of different ways, which again can be responsible for the loss of these functional characteristics which you want to have at the end. So again, we went back to the screen matrix. Here you see a slightly different overview graph rather than just a visual. Here you start to see actual cell counts for the individual wells, which I've shown you previously. And we could identify already at the beginning that there is another kind of ECM which is optimized for the growth of these cells and which is optimized for the growth of functional MSCs. So what we did was we started to look over a longer period. What happens to these MSCs? Can we grow them for extended periods of time? Can we keep expanding them? And what will we get at the end? And the test which we've run is we've run the sort of standard, which is a sort of serum-containing media versus a serum-free media. Usually conditions which are more optimized for a cell therapy. There you would have processes optimized for serum-free growth. And again, you can see that using a coating, an extracellular matrix that's specific to the expansion of cells leads to a better response than the sort of standard. So the standard serum with coating, you have a lower proliferative response versus one with a serum-free and extracellular matrix, which is specific. Another thing which we had to do was, of course, test, okay, are these cells still functional after all this time? So we looked at things like cell viability, again, their immunomodulatory capacity, and then trilineage differentiation, what you've recognized from the first example, and also included some other important aspects like the markers which they exhibit, again, telling us something about the identity of these cells. Now, the really interesting and I guess surprising thing is, why can't we use the same extracellular matrix we've identified at the beginning for isolating these cells and use them also for the expansion of these cells? Why do we have a different ECM for both? And what this picture shows you is actually a suboptimal growth type. So what you're seeing here is MSCs which have been seeded on isomatrix, and they don't expand as well as cells which are seeded on the expansion matrix. So what does that tell us? It tells us that there are niches within the body. I guess this is a known concept. There are niches within the body which are responsible for certain jobs. And evidently from what our product development has shown, you have a specific ECM for sort of maintenance of your stem cells. That would be sort of your bone marrow. And I guess you have another kind of ECM which is responsible for rapid proliferation of these cells. So this is not the same thing. They are two very, very different things. Now, what does that mean then for cell manufacturing in total? We think that the implications for this are really, really exciting. So in particularly for patients which are relying on these cells for a particular treatment. So what you're seeing here is a comparison of a sort of standard isolation using serum and expansion versus a expansion using either the isomatrix or a combination of the isomatrix and our expansion matrix. And these differences are on a logarithmic scale, so they're really massive, right? Even though these lines appear to be close together, you have something like 10 times more cells manufactured using the sort of optimized method. So what we have essentially created here is a way to isolate more stem cells from each individual patient, and not only that, expand more cells from each individual isolation that is made, right? So the implications for final patients could be very, very large. From the data from this paper, I believe we've isolated from more than 14 patients. The indication is that there would be a benefit of at least 30% more stem cells over the entirety of the project. So that means that if you had sufficient stem cells to treat 100 patients, at the worst case scenario, you could deliver treatments to 130 patients, right? So this would be, significant for a cell therapy manufacturer. So maybe I'll just talk about sort of the next step, what happens after that, and that is where we're speaking about scale up, so going beyond characterizing the cells and seeing how they expand in the laboratory surrounding, but looking at what does it look like when you're expanding these cells in a manufacturing background. So we work together with a company called Rooster Bio based in the US, very well known in the cell therapy space for MSC manufacturing. And they've generated this data using essentially the extracellular matrix coatings we provide them for the expansion of these cells on microcarriers. So what you see in the lower right-hand corner are these beads. Essentially, these are polystyrene microcarriers, approximately 200 microns in diameter, which are then coated with our specific ECM and are used then in a bioreactor setup to massively expand cells. And here you're looking at not millions, you're looking to generate almost billions of cells since you want to treat many, many patients, right? And what they've come to conclude is that with various donors and also various sources of MSCs, you're able to achieve clinically relevant quantities of MSCs using their media and particularly our extracellular matrix coatings. So yeah, this has been a deep dive really into MSCs, but we've used the same technology to tailor extracellular matrix for a wide variety of different cell types, both human and animal. And essentially, this is sort of the call to anybody who's interested, is that we could start tailoring with our technology for whatever research question, process, or cell type that you have of interest. And it's something which we can do as a sort of service. We have done that with everything from all of these different cell types, so everything from brain to heart to gut, skin. So it's been very, very broad. And maybe I would just conclude with an overview of some of our product lines. So we've developed our first product in the MSC space. I've mentioned at length during today's talk the isomatrix, the expansion matrix, matrix MSC. But we've also developed tailored products for induced pluripotent stem cells. So that's what we call matrix iPSC, and the corresponding microcarrier products, bead matrix. That's what you've seen for our collaboration with Rooster Bio, and bead matrix plus, which is the first microcarrier of its kind on the market for iPSC expansion. With that, I would then conclude the talk, say thank you, and invite you for questions.